Hello, everyone, and welcome. My name is Brandy Davidson. I am a rising senior at Hood College in Frederick, Maryland, and I've had the pleasure this past summer to work with the Geisinger Autism and Developmental Medicine Institute, where I've utilized population genomic data to identify genomic regions unlikely to be dosage sensitive. To give a little background, the Clinical Genome Resource, otherwise known as ClinGen, is an NIH-funded centralized resource focused on identifying clinically relevant genes and variants for use in precision medicine and in research. There are four main curation activities within ClinGen located in Figure 1. This study focused on dosage sensitivity. Dosage sensitivity evaluations utilize publicly available sources, such as the scientific literature, to determine if gaining an extra copy or losing a copy of a gene or genomic region causes disease, also known as triplosensitivity and haploinsufficiency respectively. Gains and losses of genomic regions are known as copy number variants, or CNVs, and information about the dosage sensitivity of genes within larger CNVs can be used to assess clinical significance located in Figure 2. For example, if a deletion includes a gene with the highest haploinsufficiency score of 3, the deletion should be classified as pathogenic. On the other hand, if a deletion is approximately the same size and includes the same genomic material as a region classified as dosage sensitivity unlikely, or DSU, it can be classified as benign. This study focused on identifying those CNVs that are classified as DSU. As mentioned earlier, dosage sensitivity curations primarily rely on information published in the scientific literature, which is typically biased towards reporting those variants that do in fact cause disease. In order to identify potential DSU variants, a dataset that catalogs copy number variation in the general population, called the Database of Genomic Variants, or DGV for short, was used. The idea is that if variants are found at very high frequencies in the general population, they will be unlikely to cause Mendelian disease, since those diseases are relatively rare. As shown in Figure 3, 114,555 variants from the DGV gold standard variant dataset were filtered for duplications or deletions that were larger than 1 kilobase in size and had a frequency of 5% or greater, with more than 2,000 alleles tested. Out of the 15,941 CNVs that met this criteria, only 220 of the variants included at least one gene. These 220 variants included about 750 genes, which were then manually reviewed using the literature, OMEM, and ClinGen in order to exclude any with potential disease associations. After this assessment, 184 potential DSU variants remained. As of July 2021, ClinGen contained only 43 DSU variants. After this study, there are now 227 DSU variants, a near 430% increase as shown in Figure 4. These variants will be made publicly available through clinicalgenome.org. Population data can be used to identify DSU variants, allowing clinical laboratories and medical professionals to efficiently filter variants unlikely to cause disease. Resources such as this allow the genomics community to focus on the variants that may be relevant to their patients.